Hello, everyone, and welcome to the Precision Medicine virtual event. My name is Matthew Bainbridge. I'm the Associate Director of Clinical Genomics at Rady Children's Institute for Genomic Medicine, and I'll be presenting on precision pediatric genomic medicine. Please feel free to submit any questions during my presentation, and I'll follow up with you via email. So I think the first question that we really have to answer is, what is precision genomic medicine? And I have a nice short video to help demonstrate that. So if we can just play the first video, please. This little girl is 1 year old. A 1-year-old should be able to sit up unaided, should be able to walk, and certainly should be able to reach for a toy. But this little girl cannot. She can follow the toy with her eyes. You can see she can move her mouth, but she can't reach for it. She's effectively paralyzed. So this little girl came into our neurology clinic, and we sequenced her, and in 3 days we got a provisional diagnosis. And what we actually found were biallelic mutations in a gene called TH. So what does tyrosine hydroxylase do? Well, it takes tyrosine, hydroxylases it, and turns it into L-DOPA, and L-DOPA is then turned into dopamine. And if you don't have any TH, you can't make L-DOPA and you can't make dopamine, and dopamine is an extremely important neurotransmitter. So this girl just could not make this neurotransmitter, and this is the cause of her symptoms. Very fortunately, we can artificially synthesize L-DOPA, and we can give L-DOPA to her as a pill. And that allows her to actually bypass the block at tyrosine hydroxylase and produce her own dopamine. And so here you can see in this next video what it looks like after she's been on L-DOPA treatment for about 4 months. So here we have that same little girl, and this is after 4 months of treatment. You can clearly see that she's sitting up very well. You can see that there's still a little bit of deviation in her ankles, but she can grab toys and move around unaided and hold her head unaided after just 4 months of treatment. That's Dad there. Dad got Dad to lay her down, see if she can get up, and she can get up just fine. That's Big Sister. So she can get up fine on her own. She can reach for toys. Okay, now we're going to get Dad to lay her down on her front. And you can see she actually has so much strength she can really resist being laid down on her front. And she can easily hold herself up. This girl basically went from being paralyzed to being almost fully functional in 4 months. That video is about 1 year old now. If you saw that girl walking down the street, you would not know anything was wrong with her. She's perfectly fine. She can run, she can walk, she's going to develop normally and live a happy, healthy life. And this is really what we mean by precision genomic medicine, is that we go in, we sequence these kids, we figure out exactly what's wrong with them, and then we have an important and efficacious treatment that can make their lives better. So this is what the current state of the art looks like in the ICU, children's ICU. About 55% of the kids come in with an unknown disease etiology, and we can actually sequence them in sort of 1 to 2 days. And while they're getting that sequencing, they get interim empirical treatment. We make a diagnosis in 20 to 46% of cases, depending on the cohort. And of those, about 31% overall can have some sort of change in management, and then 21% overall have a change in outcome. There are 3 very important steps to actually making this work. The first is speed. It's very important that we can get these diagnoses and treatments to these kids in a timely fashion while there's still some chance to actually make their lives better. The second is accuracy, and by this we mean we need to know actually that the diagnosis they get is correct. That when we call a mutation pathogenic, it is pathogenic. And the last thing, of course, is treatment. There's no point in being able to do all of this if we can't actually make some change to the child's health. So I'll be talking about these 3 factors and what we're doing at Rady Children's to influence them. So the first thing I'll talk to you about is speed. Speed is extremely important for acutely ill children. A non-rapid diagnosis is clinically irrelevant for them. We need to be able to treat these diseases, and we need to be able to treat them early. Realistically, we can deliver this now in 1 to 3 days, but ideally we'd be able to actually return this in less than 8 hours so that a child would be admitted and during the same shift, we'd actually have a genomic diagnosis for them. Realistically, this is only possible by basically pre-sequencing all children at birth. That would be like doing it as a newborn screen, and then we could just do analysis after those kids are entered into the NICU. This is a handful of the kids that we've made diagnoses on at the Institute. All of these diagnoses have an effective treatment. Some of them are very expensive, they're like gene therapy. Some are very inexpensive, like getting vitamin B6 supplementation or getting galactose. What you can see is that all the kids at the top, all those kids that we sequence later in life, have either died or been neurologically devastated by their disease. Whereas generally, if we can sequence those kids below 4 months of age, or preferably below a week of age, they actually do well and develop normally. About a year ago, we sequenced the 19-hour genomes. That means we went from a blood spot to a diagnosis in 19 hours. So this is just regular for us now, just to be able to turn out a genome that quickly. I'm actually going to concentrate primarily on the bioinformatics steps after the initial sequencing, because that's where our focus has been for the last year. So a standard diagnostic interpretation can take anywhere from a day to 2. It takes about 18 hours for us to produce all 125 billion bases of sequencing data, and it takes about an hour for us to align that data to the genome and then call the variants. What can be really time-consuming and very manual is doing the interpretation of that data, and that can take anywhere from 7 to 12 hours, because really what we need to do is we need to whittle down those thousands and thousands of variants to the 1 or 2 that are actually causative. One of the key things in being able to do this is being able to prioritize those variants based on the disease phenotype. And the way we do that is that we gather what are called HPO terms. So this is the human phenotype ontology. And this is a set of standardized terms as well as their relationship to other terms that also describe phenotypes. So a cardiac phenotype might be subdivided into very specific kinds of cardiac phenotypes. And this allows you to do fuzzy matching. So you don't have to perfectly put in a phenotype for a child. You can put in something that is approximately correct, and we can then figure out how to use that data usefully through fuzzy matching. And really what we try to do is that we take the ontology terms for every disease, and we compare it to the ontology terms that the child has, and we do an overlap, a fuzzy matching, and get a score to say how much this child resembles a particular disease. And this then allows us to rank the variants that the child has based on their phenotypic overlap to known diseases. Part of the difficulty in doing this, though, and that's a very quick process and it's very well established, but part of the difficulty is actually pulling that data out of the EMR. So an EMR, for acutely children, they're usually smaller, but for some of our chronic children, you can have hundreds and hundreds of documents. And sometimes very important phenotypes can be buried in the EMR. They're lost. So the child could have had a MRI when he was 2, and the results from that MRI may not be continue on in the EMR. So you may not pick it up by just looking at sort of the last couple of weeks of EMR data. So we worked with a company called Alexion to generate a program called CliniThink, and CliniThink goes into the EMR and extracts HPO terms using natural language processing. A process that could take 30 minutes to several hours can be accomplished in about 20 seconds. And CliniThink actually is very, very good at pulling out meaningful terms and generating good HPO terms from the EMR. Using that data, we can then, like I said earlier, overlap the child's phenotypes to the known phenotypes are caused by disease mutations. And that allows us really to sort of whittle down from, 1 million mutations down to the one causative mutation in about 5 minutes. The next thing I want to talk to you about is accuracy. And accuracy can mean a lot of different things to different people, but really what I'm interested in is when we call something pathogenic or when we call something benign, are we actually correct? And this is really highlighted by what they call the variant of unknown significance problem, or the VUS problem. VUSes are when we don't know a variant is pathogenic or not. They're a real problem because they actually impede a child from getting a diagnosis. Generally, clinicians will almost always treat this as benign and basically continue the search to get an actual diagnosis. Interestingly enough, it does not affect all genes equally. So ASXL3, which is on the left there, this gene, it's dominant. It's caused only by de novo mutations. It's very severe. It has high penetrance, high severity, and consequently, we're very good at knowing whether the mutations in this gene are pathogenic or likely pathogenic. When you take a gene like KCNQ2, it can be inherited. It has variable penetrance. And so we're less sure necessarily whether we can call a variant pathogenic or likely pathogenic. RYR2 causes arrhythmias. It can be inherited. It has variable penetrance. It has variable expressivity. And it's also a very, very large gene. And subsequently, over half the mutations in ClinVar are VUSes for RYR2. So how can you resolve a VUS? Well, if you see the same variant over and over again in independent families, and they all have the disease, and the variant's rare, so we don't see it in lots of unaffected individual, we can probably implicate it as being pathogenic. The other thing you can do is it can do experimentation in a model system. And there's tons of animal models like mice, fish, frog, flies, chickens, and they're great because they can actually phenocopy the disease. So if you have a child with seizures and you put the mutation into a mouse, the mouse might have seizures as well. You can also get multiple phenotype readouts, which can be really advantageous. So a mouse might have seizures, but they also might have impaired balance and movement. They might act strangely in an open field test. There's lots of things you can measure when you're looking at an animal model system. And once you have them generated, they tend to be fairly easy to maintain. On the con side is that they can be expensive to generate, sort of $5,000 to $15,000. And more importantly to us, it can be very time consuming to generate these. So it can take 6 months even to make a fish model, 18 months plus to make a good mouse model. When we compare this to a clinical turnaround time of 3 days, that's really not acceptable because the research turnaround time can be months and months to years and years. And it can actually be so long that result may never actually get back to the patient because the patient has moved on and gone to a different provider. You can use things like cellular model systems, which are single cells or sometimes organoids. They're faster to do, they're less expensive, and tend to be easier to manipulate genetically. However, they can be difficult to maintain. And generally, you can only really pull out a cellular phenotype. So on the left here, that's a patch clamp. So we're looking at the electric potential across the membrane of that cell. And that can be fine for a lot of phenotypes, but sometimes it's not. And sometimes it actually does not replicate what we see in the clinic. That is, it doesn't look pathogenic when you're just measuring the cellular potentials, but it is pathogenic because the children actually get sick. I'm going to talk about worm as a model system. So this is C. elegans. It's a small 1 millimeter invertebrate multi-cellular organism. It has some of the best understood neuronal development of any organism. It's an inexpensive model system, and we like it because it's a compromise between complex, expensive mouse models and inexpensive cellular models. And so it still has some features of both. And this is a collaboration that we're doing with Metrics, which is a company in Eugene, Oregon. It's worth taking a moment to talk about phenologs and phenocopying. Generally, as we get more and more distant from humans as a model system, we're less able to perfectly replicate the phenotype. So on the right there is a fish, and on the top that is a control fish, and on the bottom that is a fish with a cardiac defect. And this cardiac defect, rather than measuring, say, ejection fraction or the thickness of the cardiac wall, which we would in patients, what we're doing is that we're really just looking at the edema as a readout. So that's the swelling that you can see there indicated by the arrow. Even when you have mouse models, they may not actually be able to replicate the phenotype that you see in the patient. So those are 3 common diseases that are well understood. And when you knock the equivalent gene out in the mouse actually doesn't get sick. But you can read other readings to actually see whether those mutations are pathogenic or not. And so what we talk about when we're looking at something else, rather than a phenocopy, is what we call a phenolog. So it's an equivalent or a proxy for a clinical phenotype. Worms don't have brains or hearts. So we have to look for phenologs in something else. And as it turns out, the orthologous genes that we have in our brains and our hearts are actually expressed in the mouth and GI tract of a worm. And in fact, 50% of human genes have a good worm ortholog, but actually 70% of human disease genes have a good worm ortholog. And this really speaks to how important conservation is to disease genes, or vice versa. Even if you do find a good modeling target, so even if there is an ortholog, you may not actually have sequence conservation. So that's another thing to worry about, especially when you're really interested in inserting VUSes into these genes, because you need to have actually perfect sequence conservation. And the way we get around that is by humanizing the worm. Once you have your target gene, what you do is that you actually just directly synthesize the cDNA, the human cDNA, and then you add to it on either side the flanking gene regions of the worm ortholog, and you insert this in a plasmid. And the reason why you do this is because when the plasmid can then recombine with the worm, and it actually knocks out the worm gene and replaces it with the human gene. And this is advantageous because this gene, this human gene, is actually under control, the same genetic controls as the worm gene would be. So it will be expressed at the same time, it'll be expressed in the same organs, it'll be expressed at the same level, and it'll respond to stresses in the same way. Making the transgenic involves a couple of steps. So the first thing we do is that we'll take a wild type nematode and knock out the gene. And hopefully we get a phenotype. So we can measure the phenotype on the knockout. Then what we do is that we take a wild type worm, we inject that plasmid that we generated that has the human ortholog, and we humanize the worm. And what we look for here is a knockout rescue. So we want to see a different phenotype than we saw in the knockout. It doesn't necessarily replicate the wild type phenotype, and I'll give you a couple slides in that in just a minute to show you what I mean by that, but it needs to be different from the knockout. Once you have this generated, you can actually use CRISPR to go and put in the variants of interest, those variants that you want to test, and then you measure the phenotype. And the phenotypes you can measure on worms are things like movement and growth and reproduction. You can look at morphology, you can look at feeding. What's very interesting about this is that humanizing that worm is a fairly lengthy process. It can take sort of 2-3 months. But once you have that humanized worm, CRISPRing in those variants of interest is actually very fast and can be done in as little as 10 days. And this is actually a really clinically relevant time frame. It's not obviously as fast as being able to genome sequence the worm in a day, or genome sequence the patient in a day, but it does actually can get you to understanding a variant of unknown significance in 2 weeks. We want to explore neurological diseases as our model because they're fairly common. They affect a large number of the population, and they're expensive. It's about $30 billion annually. They can also be difficult to model or quantify in vitro. So in vitro models do not always do a good job, especially replicating movement disorders. But also, really importantly, there's actually 28 anti-epileptic drugs available in the USA, which means if we can make a diagnosis for a kiddo, there's possible treatments for us to give him. We're going to concentrate on a gene called CACNB4. It's widely expressed in the brain. It mediates the influx of calcium ions into the cell. And really what it does is that it alters the activation and inactivation potentials of the cell. So we have 4 variants. We have a frameshift variant that's associated with epilepsy. And we have one of the most common variants, the C104F, which is one of the first identified variants in this gene. And it's associated with episodic ataxia in a French-Canadian cohort. We also need to test a benign mutation to make sure that we can tell pathogenics from benigns. And then we have this last mutation, which we call hyper. It's actually an insertion deletion. So it's a deletion of 3 amino acids, HYP, and an insertion of arginine. And this is a variant of unknown significance. So this was in a patient that we sequenced that has an ataxia and ptosis phenotype. So the first thing we do is that we knock this ortholog out in the worm, and that kills the worm. They arrest in a larval stage, early larval stage. If we insert the human version of the gene, we actually see that the worms do not arrest, and they grow up and they're fine and they live happy lives. Next thing we want to do is measure some phenotypes. And one of the phenotypes that's very good at being measured is the electrophysiology of the feeding worm. So what you see up here is what they call the N2. This is a wild type worm. And this trace looks a lot like an EKG, but worms don't have hearts. What we're seeing is the rhythmic feeding of the worm's mouth. These 2 traces are the same trace, just one is longer than the other. So the trace on the left is about 5 seconds and the trace on the right is about 20 seconds. So this is what a normal feeding worm looks like. The knockout obviously is flatline because the knockout is dead. This is what happens when we put in the human CAC NB4. We can see that it doesn't return to wild type, that the worm is alive, but he actually feeds slower and more sporadically. So you can see on the right that the feeding seems to come in bursts. What we actually think is happening is that this gene is working sort of too well and actually sort of shutting down the neurons and not letting them fire as quickly as they would like to normally. When we introduce this known causative pathogenic mutation, what we see is something kind of interesting, that is actually returning to something that's a bit more like wild type. And what we think is happening is that this mutation is damaging the function of this gene without turning it off. And so it's allowing the neurons to actually fire more quickly. And then when we look at the VUS, what we see is something that is, again, very similar to wild type and similar to the known pathogenic mutation. This is another way of visualizing that data. So we're measuring the frequency of eating on the left. And you can see that there's a not significant difference between the humanized version and the benign over there on the right in green. And there is a significant difference in the mean feeding frequency between the known pathogenic and our VUS mutation. You can see the same sort of data there on the right, but it looks a little more dramatic. This is a cumulative distribution plot, so it goes up to 100%. And you can see that our VUS and the known pathogenic mutation clusters very, very closely together, whereas the benign mutation does not. You can measure multiple things in these worms, like I said, not just feeding, but we can also look at chemotaxis. So what happens there is that you put the worms in a little petri dish and you put a chemo attractant to one end and you give them 30 minutes and see how many of the worms actually get over there. ODR10, which is in the center there, is our negative control. So these worms cannot sense the chemo attractant. And interestingly enough, the human knock-in actually replicates the wild type phenotype. So it does not greatly affect the worm's ability to move. And nor does the pathogenic C104F, but interestingly enough, the hypermutation does, and the worms actually seem to move a little bit faster with the hypermutation. So the real question is, after doing all of this, is hyperpathogenic? And the answer is we still don't really know. It's clusters with pathogenic mutations, but we don't have enough data really to be able to tell whether it's pathogenic or not. And this is work that we're going to be doing in the next few months, is just really nailing this down. And it's quite important because people with that C104F mutation can be treated by a drug called Diamox. And that brings us to treatments. So like I said before, being able to do this and make these diagnoses and determine whether the VUSs are pathogenic or not is great, but if there's no treatments, it's not especially important to the outcome of these children. There's three kinds of treatment types. Starting at the bottom, most people are probably familiar with drug discovery, which is where you take huge libraries of tiny molecules and try to find a brand new drug for a disease. But there's also drug repurposing, and that's where you take a drug that has had some or possibly a lot of clinical trials and clinical data, and might actually be approved for a particular disease. But we're going to go and repurpose it for a different disease. And then right at the top is what I call regimen discovery. And this is useful when you have a bunch of drugs for a particular disease available, but you don't know what the right one is to give to the child. So having made a worm with a particular child's mutation in it, you can then use that worm essentially as a therapeutic avatar, right? And what we want to do there is that we want to take, say, the 28 anti-epileptic drugs that are available in the US market, and rather than trying each one on the child until you find something that works and then stopping there, so you try all 28 in parallel on the worms, find out which ones work the best, and prioritize trying those on the child. So let's experiment on the worms rather than on the children. And this is, you can also think of it as phenomimicking or phenocopying a drug response. So this is also work that we're going to be doing in the next few months on CACNB4, as well as another gene called KCNQ2, which is a common epilepsy gene. We are also working in close conjunction with a group called Caliber, and Caliber is part of Scripps Research. And this is one of those companies that can do massive, massive high throughput screening of hundreds of thousands of molecules. But our interest is actually in repurposing drugs. So they actually have a library of 13,000 compounds that have some kind of clinical data already. So a lot of them finished stage 1 clinical trials, and they call this the Reframe data set. And what we do here is that we introduce the mutations into a cell. or the cell can give us some phenotype that can be read through in a high throughput manner. We then use these massive libraries and a whole bunch of rather complex robotics to go and rapidly screen through all these different drugs to look for a drug that can be repurposed to alleviate the phenotype. And for this one, we're actually working on a different gene called ADCY5. It's a movement disorder gene. And the reason why we like it is that it's actually a gain of function gene. And so we think generally when we have a drug binding to a gene, we're going to, or to a protein rather, we're going to be turning that protein off or turning it down. And so really we're interested in these gain of function mutations as good targets for initial screens. So this is what we envision the future of precision pediatric medicine to be like. These kids come in, we don't know what's wrong with over half of them. They've already been sequenced because they were sequenced in the first week of life. So all we have to do is pull in their sequence data and use natural language processing to get their phenotype. And then in 5 minutes, we can go and prioritize the most important mutations. Potentially, we have a definitive diagnosis at that point, in which case we can modify the treatment and hopefully discharge the child. If we don't, because we don't have a good mutation, we can't tell it's pathogenic, we can go and rapidly introduce that VUS into a worm model system, and in 15 days, hopefully we can have a definitive diagnosis. If there isn't a known treatment or there's too many treatments to pick from, what we can actually do is that we can then go and do drug regimen discovery using that worm that we've just made to test the VUS. So I would like to thank Stephen Kingsmore, who's the CEO of Rady Children's, David Dimmick, who's our Chief Medical Officer, Shamul Chowdhury, who's our lab director, and Jenny Friedman. Many of these patients I've talked about are hers. At Nemometrics, I'd like to thank Chris Hopkins, who's their CSO, as well as Shula Jaran, who's their Vice President of Business Development. And lastly, at Caliber, I'd like to thank Nikki Alvarez, who's their Associate Director of Alliances.